MCAT 2017 Cram, Biochemical and Biological Foundations of Living Systems. Passage 20, Cancer and Hexokinase. As you view the reading of the passage, you'll notice some highlighted snippets of text. What I want you to do is garner meaning from these specific selections in order to answer the questions that follow. The passage is extremely manageable, and so are the questions, so good luck and happy reading. Paragraph 1. A key component of many cancers, especially the most aggressive, is their ability to metabolize glucose at a highly elevated rate. The first step in the metabolism of glucose is its phosphorylation to form glucose 6-phosphate, which traps the molecule within the cell. As shown here, glucose entering the cell through the GLUT4 receptor and being converted to glucose 6-phosphate via the hexokinase enzyme. This reaction is unable to proceed in vitro unless the protein or enzyme, hexokinase, and magnesium cations are present. The concentration of hexokinase and magnesium cations remain constant throughout the reaction. Paragraph 2. Different forms of hexokinase, that is, hexokinase 1, 2, 3, and 4, are found in differing levels throughout the body. Each form displays unique functional characteristics. Figure 1. Saturation curves for hexokinase 1 and hexokinase 4. So on the abscissa, we have the concentration of glucose in millimoles per liter, so the millimolar concentration. And on the ordinate, we have arbitrary enzyme units ranging from 0 to 100%, or if you look at them as fractions, from 0 to 1. All right? Okay. Figure 2. In vitro kinetic parameters for hexokinase 1, 2, and 3. So here we see that the Km is lowest for hexokinase 3 as it relates to the glucose concentration. And then we see for ATP, the Km is lowest for hexokinase 1. Okay. Note that values shown are representative of scientific literature, and values for hexokinase 4 are not normally reported. All right? Paragraph 3. Tumors can be located using positron emission tomography, PET for short, to assay or determine hexokinase activity levels. 18F 2-fluoro-2-deoxy D-glucose, 18F FDG for short, is a radiolabeled form of glucose that can be taken up normally by all cells and phosphorylated by hexokinase. The 18F isotope undergoes positron emission, allowing it to be detected by PET scans. Since 18F isotope replaces the 2' hydroxyl group, it also prevents further metabolism of the molecule. After the intravenous administration of 18F FDG, a whole body PET scan is carried out to measure the radioactivity levels in various tissues of the body. All right? Studies have shown that 75% of 18F FDG injected intravenously enters the cell and undergoes radioactive decay with a half-life of 120 minutes. 25% does not enter the cell and is excreted with a half-life of approximately 15 minutes. What percent of 18F labeled molecules originally injected are still radio-labeled and present in the body after 2 hours? Is it A, 25%, B, 38%, C, 50%, or D, 63%? I'll give you a moment to decide. All right. Okay, so this is a half-life problem with two distinct groups of molecules. The first group of 18 FDG enters the cell and has a half-life of 120 minutes. The second group of 18 FDG remains in the bloodstream and has a half-life of 15 minutes. In 2 hours, the 18 FDG in the cell undergoes one half-life, okay? Since you began with 75%, half of that remaining would be approximately 38%, but let's take a look at the calculation, okay? Alternatively, another way of looking at 75% is 3/4 of a sample, and half of that would be 3/8, thus 38%. Again, so 3/4 of the samples that enters the cell times 1/2 raised to the first power, we don't have to write the first power here because that would be redundant, is going to give you 3/8. Now, if you think of this in terms of 100%, 100 divided by 8 is approximately 12.5. So there's 8 units of 12.5% in 100%, and 3 times 12.5 is going to yield 37.5. That's why the final answer is going to be 38%. But let's see why as well in terms of the second portion of 18F FDG that remains in the bloodstream. Okay, the remaining 25% undergoes 8 half-lives because there are 8 15-minute periods in 2 hours. If you begin with 25% or 1/4 of an entire portion, you can quickly see that much less than 1% is going to remain. So 1/4 is our starting portion with the amount of 18F FDG that remains in the bloodstream times 1/2 raised to the 8th power. So it's going to be 1/2 times 1/2 times 1/2. This dwindles down to next to nothing. So we can ignore this group of molecules because it's essentially completely excreted, okay? And since only the 18F FDG in the cell remains to an appreciable amount, we can say that approximately 38% of the radio-labeled molecules are still in the body. Why are Km values for hexokinase 4 not reported in the scientific literature? Is it A, hexokinase 4 Vmax decreases with increasing glucose concentration? Is it B, hexokinase 4 Vmax remains constant at physiological glucose concentrations? Is it C, hexokinase 4 glucose affinity changes as glucose concentration changes? Or is it D, hexokinase 4 glucose affinity remains constant at physiological glucose concentrations? I'll give you a moment to think this one out. Definitely refer back to the passage if you need to. All right. Since hexokinase 4 is not included in figure 2, only hexokinases 1 through 3 are, the main source of information from this particular passage without any outside knowledge is going to be garnered from figure 1. Okay? So notice that the curve for hexokinase 4, the red curve, is S or sigmoid shape, suggesting cooperativity. Cooperativity is when the binding of the substance to the enzyme facilitates the binding of more substrate, okay? So similar to a positive feedback mechanism or cascade. In other words, as the concentration of glucose changes, the affinity of the enzyme for glucose increases, okay? So an enzyme that displays cooperativity does not follow Michaelis-Menten kinetics and doesn't even have a Km value to begin with. So if it doesn't have a Km value, you can't report it in the literature because it doesn't exist. Therefore, the reason why hexokinase 4 doesn't have a Km value that's reported is because hexokinase 4 glucose affinity changes as the concentration of glucose changes. All right? Okay. What can Mg2 plus, the magnesium cation, be considered in the hexokinase reaction? Okay, what can it be considered? Is it A, a cofactor, B, a reactant, C, a product, or D, an enzyme? You decide. I'll give you a moment to think. All right. Okay, so we're told in the passage that the concentration of magnesium cations is the same before and after the reaction, so it's neither a product nor a reactant, okay, or vice versa. And enzymes, by definition, are organic catalysts, organic protein catalysts. And the magnesium cation is definitely an inorganic ion, so it doesn't fit the description of an enzyme. By default, the correct answer choice is answer choice A. The magnesium cation is not an enzyme, but it is clearly required for the proper functioning of the enzyme. Therefore, it's an inorganic cofactor. Okay? And as it turns out, the magnesium cation associates with the negatively charged triphosphate group of ATP to help hexokinase facilitate the transfer of a phosphate anion. Okay? All right. Normal blood glucose levels typically range from approximately 5 millimolar per liter to 10 millimolar per liter. Which form of hexokinase is least likely to be found at elevated levels in a cancer cell? Is it going to be A, hexokinase 1, B, hexokinase 2, C, hexokinase 3, and you guessed it, D, hexokinase 4? I'll give you a moment to think and definitely refer back to the passage if you need to. All right. Okay, so we know from the passage that cancer cells metabolize glucose at a highly elevated rate, okay? Therefore, the hexokinase with the lowest affinity for glucose is the least likely to be found in cancer, or in a type of cancer rather. So from figure 1, we see that hexokinase 1 has a much lower Km value, and hence it's more active than hexokinase 4 at physiologic blood glucose levels. Okay, so we're in the range, let's see, from 5 to 10 millimolar. Yeah, so between 5 and 10 millimolar, it's much more active, hexokinase 1. Therefore, hexokinase 4 is less likely to be expressed in a cancer cell characterized by elevated glucose metabolism. Okay, so out of these two, hexokinase 4 has less of a chance of being expressed. Then, if you take a look at figure 2, we can compare the Km values of hexokinases 1 through 3. All right? The Km value is the glucose concentration at which the enzyme is operating at half of its maximum activity, Vmax. Let me just write that out for you, Vmax. Or maybe not. Yeah, just think about it in your head. Okay, yeah, so that's that, Vmax. The lower the Km value, when it's like really, really tiny, the higher the affinity of the enzyme for this substrate because it doesn't take a lot, a lot of substrate to reach Vmax or the maximum operating rate. Okay, so hexokinase 3 out of these three is definitely going to have an even higher affinity for glucose than hexokinase 1. Okay, and although hexokinase 2 Km is low, it's still not lower than these two, but it is much lower than hexokinase 4 technically doesn't have a Km. It's still very close to these two, so we're not going to assume that it has a slower operating rate than hexokinase 4, okay? All right, because all of the Km values are less than 1, so yeah, it's not less than these two, but it's still pretty good, especially when you compare it to hexokinase 4. Thus, hexokinase 4 is by far the least active of these enzymes. This is a strong conclusion or assumption that we can make based on the data provided and is therefore least likely to be found in elevated levels in a cancer cell. All right? Okay. Which molecule is formed when hexokinase acts on 18-fluoro-FDG? Is it going to be molecule A, molecule B, molecule C, or molecule D? I'll give you a moment to decide. All right. Okay, so from the passage, we know that the 18F isotope replaces the 2' oxygen that would be located in the glucose ring. Therefore, we should see 18F and not oxygen 18F. Okay, the fluorine 18 isotope alone, that's it. And from the passage, we also know that hexokinase phosphorylates glucose at the 6' position, which is right here. This would be the 6' oxygen because we form glucose 6-phosphate as opposed to glucose 5-phosphate or glucose 4-phosphate. Okay, so the only structure that has both of these features, the fluorine 18 isotope and phosphorylation occurring at the 6' position, is going to be structure C. So answer choice C is the correct answer choice. Okay? All right.